In this video, we'll just get started talking about the mineral nutrition of plants, and specifically in this video, we'll talk more about nutrient uptake. So, two aspects that we're going to talk about when we're talking about mineral nutrition of plants is, first of all, how do they get the nutrients, and then how do they use them? So again, this video will be focused on number one. For number two, we'll do an activity in class and talk more about how plants are utilizing the nutrients once they have them. All right, so first, just a couple things about the variability in nutrient availability to plants. And so you would get much more details on this if you take a soil science or soil physics class. So this will just be a very brief overview. But one of the things that happens in soils, or something that creates variability in nutrient availability, is the age of the soil. And two of the most limiting nutrients globally are nitrogen and phosphorus. And so I'm going to focus on those largely throughout the whole video, and this figure here also focuses on those. So what you see is kind of the nitrogen and phosphorus availability are kind of inversely related, at least in relatively young soils. I mean, these are still really old soils, but anyway, in very young soils, you have lots of phosphorus availability. So a lot of times in volcanic, new soils formed through volcanic activity will have really high phosphorus availability, but that just gradually decreases over time. And so like our tropical soils, which are some of our oldest soils globally, are really low in phosphorus availability. And that's partly because phosphorus isn't regenerated or put back into the soil in any way. So as it's used up and weathered, it just slowly disappears, I guess, for lack of a better word. Nitrogen availability, you can see, is different. In very young soils, there's not a lot of nitrogen, but as the biological activity occurs, you get more and more nitrogen building up in the system. But then as soils get really old, they lose their capacity to hold on to that nutrients or to the nitrogen, and that also, that availability of nitrogen also decreases over time. So that's one thing that can cause variability as we look around the globe. Another one that's having a bigger impact now on nutrient availability is deposition of different nutrients. So this map is just for phosphorus deposition. And you can see the yellow areas are higher phosphorus deposition, and then greener a little bit lower, and then as you get to the darker blue, that's the lowest levels. If we were to look at a map of nitrogen deposition, it would look very different. It would have different areas of high input, but I just want to highlight this because this is causing changes in nutrient availability, and not just for nitrogen, because we talk about nitrogen a lot, or I feel like it's talked about more in the media, but phosphorus deposition is also impacting nutrient availability. And then last thing I just want to mention is that specifically, or most commonly for nitrogen, plants can use different forms of nitrogen. And we'll talk more about nitrate, which is the NO3, and then ammonium, which is NH4. Those are the two forms of nitrogen we think of being, plants utilizing. But more recently, people have realized and discovered that plants can actually uptake specific amino acids. So this is glycine, which is an amino acid. So before this, we thought plants had to produce all their own amino acids from those basic building blocks, but they can actually take up some of those as well. So that can cause variability in the availability of certain nutrients for certain species. So if a species can't uptake glycine, that's great, they can use that form, but some species aren't capable of taking that up. So that's something else that on a species level can change how many nutrients are available to them. So just some general things to think about in setting up this conversation of, how nutrients are varying over time. And so then we're going to talk about, how plants deal with that and can take up nutrients despite that variability. So the question is, what is the major driver of nutrient uptake rate for individual plants? So we're talking about a plant trying to get nutrients out of the soil. What's controlling its rate of uptake? And so despite some of the variability we just discussed, the primary controls on nutrient uptake of individual plants occurs in the rhizosphere. So this is basically the part that the plants can control, right? They can't control necessarily how old the soil is or what deposition is happening, but this is allowing them to control, some aspect of nutrient uptake. And so that rhizosphere is the portion of the soil that is influenced by plant roots themselves. It's vague intentionally, because it's really hard to create a definitive boundary around the root system where it has an influence. But there's some zone around the root system where those plants has some influence in that area. You get too far away from the root, and the root really isn't having a big impact on that area. So that's what I mean by the rhizosphere. Okay, let's talk about nutrients in the soil briefly before we talk about them getting into the root. So most of the nutrients that we're going to be focused on are going to be adhered to the soil particles themselves. So many of the nutrients that we're going to look at have positive charges, so they're cations. So some of the common ones are shown here: calcium, potassium, magnesium. Ammonium is another one that's not shown here that has a positive charge. And if we look at these soil particles here, it kind of shows you how many negative charges there are on the outer surface of the particle versus positive charges. And it varies a bit due to pH. So if you look at the bottom, we have pH of 3.5 to 7. This is neutral. So when we're around neutral conditions, we have more negative charges on the outside of the soil, or outside of the soil particles, I should say. As you get more lower pH values, you get more of these positive charges. And one thing I will point out, it's not here, and I couldn't find a great image of it, and I didn't want to subject you to my horrible drawings, but there are some positive charges on the interior of the soil particles. So there can be a weak positive charge of the soil particle, but generally they tend to be negative soils for at least most of the soils we will work with. So soils generally have negative charge. That means the cations, or the nutrients with positive charges, are going to be adhered fairly strongly to those soil particles, and anything that has a negative charge, or anion, is going to be more loosely adhered to those particles. You can see at this pH of 5.3 there's a chlorine attached to the soil particle here. It's going to be pretty weak because there's just not a lot of spots for that to be adhered. So generally speaking, anions are more floating around in the soil, and the cations are more tightly adhered to those soil particles. This is important for the rate of movement of these ions in the soil. So there's two ways that those nutrients are moving from those soil particles to the root. The fastest one is by mass flow. So when plants are taking water up for transpiration, any soil nutrients or any ions in that solution will just be taken up in the transpiration stream. And so that's going to be the fastest way for nutrients to get into the root system. And this occurs primarily for nutrients that are held weakly or uncharged and don't adhere to the soil particles. So when we talk about nutrients held weakly, we'll talk about nitrate. So nitrate, remember, is NO3 with a negative charge. And since the soil particles tend to be negatively charged, those two aren't going to adhere to each other very well. So the nitrate is more loose in the soil, if you will, and more likely to be taken up through mass flow in the transpiration stream. This also tends to only happen when there's a lot of moisture in the soil, and when nutrient concentrations are high. When they get low, then there's just not as many of those nutrients that are going to be taken up through this pathway. This also tends to happen in the spring, so we see a flush of nutrient uptake, at least in most systems where you get, a lot of winter or spring precip. Then you see a lot of the nutrients being taken up in the transpiration stream early in the season, and then nutrient uptake slows as those soils dry out and there's fewer and fewer nutrients in the soil. After kind of the mass flow takes place, the way that nutrients are getting into the root system is mainly through diffusion. So this is a much slower process, and it occurs across a concentration gradient. And this brings us back to Fick's law again, because we're going to be kind of thinking about the flux of nutrients to the root. And so remember, the flux is equal to the conductance times a concentration gradient, or a gradient of something. In this case, we're talking about a nutrient concentration gradient. And in this case, the conductance is the diffusion rate of those nutrients in the soil. So I just have a few of them here to highlight a few things. And I didn't do any subscripting or superscripting, so bear with me. But here I've just put the relative or kind of general diffusion rates for a few ions in the soil that are important. So here again is nitrate. Remember, it's negatively charged, so it's not adhering to the soil particles. So its diffusion rate is relatively fast. So 10 to the negative 6 centimeters squared per second. Those are the units for nutrient diffusion. So 0.000006. So still pretty slow, but much faster. It's an order of magnitude faster than ammonium, which is NH4+, potassium, calcium, magnesium. All of these are in the ballpark of 10 to the minus 7, so an order of magnitude slower. And then phosphorus is the slowest, 10 to the minus 10. And we generally think of phosphorus as just not being mobile in the soil. It's like, just stuck and doesn't move, even though it does have a negative charge. So then this is kind of related to the diffusion rates, but what we kind of think of the effective diffusion distances are shown here. And what that means is how far would we expect nitrogen to be able to diffuse to a root? Or you could think about it as how close does a root have to be to one of these nutrients to actually be able to, draw it into the root system. So for nitrogen, we're talking roughly a centimeter, and that's mostly nitrate. Potassium is like 0.2 centimeters, and then you can see phosphorus, the root has to be within 0.02, which would be like 0.2 millimeters, right? So it has to be really close to phosphorus, again, because it's really immobile for the most part. Okay, that gives a little idea of the variability in how fast these nutrients might be moving within the soil. In general, the way that these plants are taking up these nutrients is by this concentration gradient. So this figure has a lot going on. Let's just focus on this dotted green line right here. This is kind of showing the concentration gradient of some different nutrients. And notice it's talking about, what's going on between 2 and 4 millimeters away from the root system. So this would, for nutrients, this would be like its rhizosphere in this distance. And so what happens is, as plants are taking up nutrients, it draws the concentration of those nutrients down at the root surface, and then leaving the concentration of those nutrients higher in the bulk soil where they're adhered to the soil particles. So we have, this is kind of, let's call this 4 millimeters away from the root up here, and down here is at the root surface, and so we have this concentration gradient. And so the nutrients, or those ions out here, are going to slowly diffuse through the moisture that's in the rhizosphere toward the root surface, and then the plants can take those up and utilize them however they want. Again, how fast that is happening is based in large part on the diffusion rate of those individual ions. So remember, phosphorus is much slower than, for example, ammonium. In this paper, I linked a paper down here where this image came from, and it just talks about like the rhizosphere and how the root interacts with different aspects of the rhizosphere. So if you're interested in that, it's a good resource. Okay, so root uptake creates this low concentration at the gradient, or low concentration at the root surface. And so the nutrients that are mobile in the soil will move from those areas of high concentration to low concentration. But if this concentration gradient isn't really like quite large, then the rate those nutrients are moving is so slow, it really doesn't support the needs of the plant. So the plant has to like have a really high concentration gradient, either because there's lots of nutrients in the soil, or it has to be able to really draw down the concentration at the soil surface. So let's talk about that a little bit. And the way plants can create that really low concentration at the surface of the root system is by using active transporters. So this uptake process isn't just passive. There are proteins embedded in the membranes of the cells at the surface of the root system that have transporters that grab onto nutrients and move them inside the root. And then the other aspect that I shouldn't have had this slide here. I had these out of order. So one way to increase the rate of uptake is those active transporters. And then we're also going to talk about root morphology. Let's just leave that. I'll come back to root morphology in a second. Let's talk more about these active transporters. So this is just a diagram from the book, figure 9.5. This is just showing the plant membrane with these active transporters. Just a couple things to notice. There are often specific transporters for cations and anions, and so the plant can kind of control what types of nutrients it's taking up. The other thing is that these can be opened and closed. So you can see this little gate shown here can open and close these, so it can prevent certain nutrients from coming in or going out, or it can, so it can just have some control of that. So specific transporters for anions and cations, it can open and close these gates. And then most importantly, well, maybe not most importantly, but importantly, this is requiring ATP. Right? So there's energy involved in these active transporters. So when plants have accumulated this carbon as this source of energy, this is one way that plants might use that energy. Some of those sugars might be transported to the roots, sugars broken back down during respiration, releasing that ATP and driving this nutrient uptake. So we tend to see lots of respiration going on when plants are actively taking up nutrients, because it needs lots of energy. The other thing I just want to point out is this is just an example of the concentration of some nutrients outside the root system versus inside the root system. And one thing I do want to show is so for some of these nutrients, you have relatively low concentration outside the root. So we're looking at calcium now, or we could look at potassium. And then we have relatively high concentration inside the root. What's important about this is that plants, because of these active transporters and their ability to kind of close and open the gates, is they can lower the concentration really low outside the root system, but have really high concentrations inside the root system. And if they didn't have active transporters, the constant, due to diffusion, those nutrients would just diffuse back out of the root and leak into the root system, and that would prevent any more uptake. But because they can kind of actively uptake and lock away these nutrients in the root system, it can just really crank down the concentration outside the root system and bring those nutrients in. So it's really important for drawing down the concentration gradient, or drawing down the concentration at the root surface. Okay, and importantly, there are, we kind of divide these active uptake or active transporters into two categories. We have a high affinity system and a low affinity system. So high affinity system means it has a high affinity for particular nutrients. And you can kind of think about that as it's really good at drawing down the concentration of those nutrients because it's so targeted. So there's specific transporters that are targeted for certain nutrients or certain types of nutrients. And so they draw down that concentration really low. So high affinity, really drawing down the concentration outside the root. Low affinity systems, on the other hand, they can't necessarily draw down the concentration very low outside the root system, but they can take up nutrients quickly. So there's trade-off between affinity and uptake rate. And this diagram also comes from the book, and I'll walk through a couple aspects of it. If you're interested in more detail, you could read the section in the book. On the x-axis here, we have the external concentration of a particular nutrient, and then we have the uptake rate. So it makes sense at low concentration, so further left here, we're going to have relatively low uptake rates, and those uptake rates are going to... Hold on, oh, my connection's lost. Here we go. I gotta switch. So uptake rates are relatively low, and then they increase as the concentration of some nutrient increases in the soil, which makes sense. A couple things to note is this I here represents the uptake rate, and then Km is the affinity for nutrient uptake. The value of Km, the way it's defined, is the minimum concentration in the soil that plants can take up nutrients. That's how Km is defined. So it's kind of weird because a low value means it has a high affinity, or it can really extract that nutrient to low concentrations in the soil. So I want to just point out a couple things here. This initial part of the curve is the high affinity system, and then what you can see here, this little break in the line. So with the high affinity system, we get some asymptote here. And then we have this break in the line and then a change in that uptake rate. This is where a plant can switch from a high affinity system to a low affinity system. So over the course of the season, as nutrient availability might change, a plant has some ability to switch which system it's using. So we would expect maybe early in the spring, when there's more nutrients available, that plant might be using a low affinity system. But as nutrient concentrations move towards the left of the x-axis, then it might need to switch to the high affinity system to be more particular about what nutrients it's taking up. You can kind of see this a little bit as well in this, this diagram on the right. And so in this system, in panel A, we're switching from a low affinity system to a high affinity system. In panel B, we're actually seeing a plant adjust its Km value. So it's almost like it can switch to the high affinity system, and then it has the ability to adjust that high affinity system or fine-tune it even more. Remember that Km is defined as the lowest concentration a plant can uptake nutrients at. So this line here is the same one as in panel A, and so this would be kind of the standard uptake system. And so in this particular system, the Km value is here on the right. It's this Km, so that's the lowest concentration it can take up nutrients. But then we have this other line here where that one kind of goes further to the left and down, and the Km value for that one is here, so it's shifted further to the left. What that means is that plant can continue to take up nutrients at lower concentrations in the soil. But you can see there's a trade-off. Its Imax value, or the maximum rate of uptake, is much lower. So the plant has some ability to continue to adjust how specific it is targeting certain nutrients. And that's mainly done by those active transporters. How many of them and what type are in the membrane? Okay, so that's kind of more from a physiological standpoint, how the plants can kind of control the uptake rates and create or maximize the concentration gradient. The other way that plants can kind of maximize uptake rate of the root system, or sorry, of nutrients, is by adjusting the root system. And this could be differences between species, or we can even see plasticity within each species and how it builds its root system. So just a couple of examples of this. In this figure here, I don't know why I put the whole figure here, but really what we want to focus on is this panel right here. And in this paper, they were looking at changes in biomass allocation of plants due to different resource limitations. And in this case, what we're looking at is root mass fraction as it varies with nutrient availability. And notice this is a relative scale, so this is really low nutrients on the left and really high nutrient availability on the right. And then the blue line is the root mass fraction that we're interested in. Root mass fraction just describes the proportion of a plant's biomass that's allocated below ground. So high root mass fraction just means most of its biomass is going to be below ground, and low root mass fraction means more of the biomass is going to be above ground. And not surprisingly, when you find plants growing in low nutrient availability, those plants tend to have high root mass fraction. So they're allocating more resources below ground to try to find and forage for those nutrients. But as nutrient availability goes up, root mass fraction goes down because there's less of a need to forage for those below ground resources. So basically just biomass allocation patterns can impact the rates of nutrient uptake. Another one is root length density. So root length density is just the length of the roots in the soil in a specific volume of soil. So on the left of this axis, low root length density means like if we were to take a core, we would just find maybe a couple of roots running through that volume of soil. Whereas the high root length density, if we had this is me holding a core of soil, we would have lots of roots running through that core, so high root length density. And then on the y-axis here, we have how much nitrogen was extracted from that patch of soil. And so, not entirely surprising, the more length of roots that's in the soil, the more nitrogen can be extracted from that patch of soil. So, not only allocating a lot of biomass below ground, but putting lots of roots, a high density of roots in the soil allows plants to really extract a lot of the nutrients in that patch. And this is kind of just reinforcing this high root length density, but this is specific to some soils in southwestern Australia, which are really low in phosphorus, and they see a lot of unique adaptations of plants to these types of soils. So each of these images are these little, this super high density of root hairs in the soil. And that's because, remember, phosphorus, I said, is effectively immobile in the soil, so it's not really diffusing to the roots at any kind of rate that can help the plants. And so plants have to go and find the phosphorus, and when they find it, they just create this burst of like root hairs growing in the area to try to extract it as much as possible. And so here's just 3 different species and 2 different scales. So they were like, 2 different scales of looking at those root hairs. So building lots of really fine roots where you find fine nutrients is a way plants can extract and speed up the uptake of nutrients. And then just driving this home one more time, this is a model of phosphorus uptake. And so they modeled all of the different aspects. You can see some variables on here we've talked about. We've got the Km value, we've got Imax, they've got some other physiological values in here, and then some morphological variables like root diameter, root elongation. And what this shows is the change in nutrient uptake with a change in each of these variables in the model. So basically this is a sensitivity analysis. So the steeper this line, the more sensitive the model is to that particular variable. And what you notice is the most sensitive parameter from the plant's perspective is root elongation. So the more roots you put in the soil, the more they can elongate and explore the soil, the faster your uptake rate of phosphorus. And this is true of other nutrients as well. So the primary driver of nutrient uptake rates is the length of the root system in the soil. It's basically just maximizing the surface area of roots in the soil and the likelihood that those roots are going to bump into an ion to be able to take up. And so even we talked about some of the more physiological mechanisms and the active transporters. Those are important, but they're secondary to how much root is in the soil. That is the primary driver as we look at differences between species and their ability to uptake nutrients. And then lastly, I just wanted to highlight legumes. I'm not going to talk much about them, but we could read some papers on them if you're interested. But legumes are a group of species or plants that have a symbiotic relationship with a particular bacteria, rhizobia. And so the bacteria can fix nitrogen from the air. So it can take N2 from the air, and our atmosphere is close to 80% nitrogen. And so there's plenty of nitrogen in the air, but plants themselves can't get it. They have to get it in the form of nitrate or ammonium or amino acids. But this bacteria can take that into gas and convert it into a biological form that then the plants can use. And so in exchange for that, the plants provide carbohydrates to the bacteria. So the bacteria don't have to go find it or eat anything else. They just fix the nitrogen and then get the nice juicy carbohydrates from the plants. And in this picture here, these little nodules that you can see, all these little balls, so to speak, on the root system, those are where the bacteria are housed. And so the plant is transporting through the phloem, is transporting carbohydrates into those nodules and then getting the nitrogen back from the bacteria. So what we see is that, when you have lots of nitrogen, you have a lower abundance of legumes. And then when you have low concentration of nitrogen in soil, you have a higher concentration of legumes. And so this is these are 4 different treatments at an experiment in Minnesota. We have just the control treatment. We have an elevated CO2 treatment. So they're flooding the air with extra CO2 to simulate future conditions. But these 2 groups on the left don't have any nitrogen added, and this is a pretty nitrogen limited system. And then the 2 treatments, the 2 groups on the right have nitrogen added. And then the legumes are these open bars. So we'll just focus on those and ignore everything else for now. So what you can see is the legumes have about 50% of the abundance or above ground cover in the plots without nitrogen, but then when you add nitrogen, the abundance of those legumes go down to around 30%. And that's because they're taking some of the energy they've harnessed through photosynthesis and giving it away to the bacteria. And so when you don't have a lot of nitrogen in the soil, that's an advantage to those plants that can do that. But when you add nitrogen, they're just giving energy away for no particular reason. They could get nitrogen from the soil relatively easily, but instead they're giving a bunch of carbohydrates away to get that nitrogen. So the more nitrogen there is in the soil, the less of an advantage having this symbiotic relationship is, and you see a decline in the abundance of those species. Okay, that's all I wanted to say about at least getting started with thinking about nutrient uptake and nutrient use of plants. Again, we'll talk more in our next class on kind of how plants are utilizing these nutrients once they get them.